folks. First of all, welcome to National Fire Biology. So in this video, I'm going to talk to you about Unit 1, KA1 cell structure. To get the most out of this video, I'd suggest you either have a jotter or a piece of paper and a pen beside you so that you can complete the activities and questions I set you as we go through. If you've completed any biology before, you should hopefully have some memory of these starter questions here. So what I want you to do is pause the video and give these your best shot. If you haven't ever done biology before, don't worry, we'll cover all of these and more within today's lesson. So the first question is draw and label an animal cell. The second is draw and label a plant cell. And the third one is to give a function of any of the parts that you were able to label. So pause, give them a try, and once you've completed the questions, we'll be ready to begin today's lesson. Perfect. So what we're going to look at just now is where we are in the course. We're currently in Unit 1 cell biology, and we're also on the first topic, so KA1 cell structure. Today we're going to learn about some of the main cell structures or organelles and their location within the cell. We're also going to learn about the basic functions of each of the organelles. Our success criteria is if you're successful, you should be able to identify the list of Nat5 organelles, briefly explain the function of each of these organelles, label the organelles within the 4 different cell types, and complete numeracy calculations on magnification and cell size. We're going to focus on thinking and numeracy as well today. So at the start of each of my lessons, I'm going to show you excerpts from the SQA's National Fire Biology Mandatory Knowledge Booklet, or Course Spec as we call it as well. This will tell you the information that can be tested in the final exam. So it has the key areas, the main parts of cell structure that we're going to look at, A and B, and it also has the depth of knowledge required. These are both the columns that can be tested. You'll be given a printed copy of these in school, but at the minute there's an online PDF version that will be available in your second task today. So the first thing we're going to look at is what is a cell. So a cell is the basic unit of life. It's the main kind of basic unit of living things. There are parts inside the cell, which you're going to learn about today, which do particular jobs, but they cannot exist independently of the cell. That's why the cell itself is the basic unit of living things. Now, the detail of the cell's structure is what we call the cell's ultrastructure. So this is what we can see if we zoom right into a cell, the parts that make it up and do particular jobs within the cell. Now, parts of the cell that carry out specialised functions and are enclosed within their own membranes are called organelles. So I'm going to use the word organelles quite a lot over the rest of these slides, and they're the bits we need to focus on and know within each cell. So what we're now going to do is at National 5, you have to be able to recognise and describe the organelles within 4 different types of cell: animal, plant, bacterial and fungal. So I'm now going to take you through each one and describe the organelles that you would find in each and how you would recognise them. What I would suggest you do is that you draw these into your notes as you go along and pause the video when needed. Okay, so this cell we have here is an animal cell. Now, the reason we know it's an animal cell is this is the most basic cell, you can see here, and it has these different parts that I'm going to point out. So the first part is the cytoplasm. So the cytoplasm always points to the white space, or what seems like the white space in the cell. On the outside, animal cells only have one circle enclosing them, one membrane, and this is called the cell membrane. Inside this animal cell, you can see this structure here. I always think it looks like a bit like the back of like a woodlouse or a bug. So this structure is the mitochondria. So mitochondria is this structure here. So here's some other ones in the cell as well. And they're always drawn roughly in this way. For number 4, it's pointing, if you can see, to these little dots. Now, some of them are in the cytoplasm and some of them are attached to this. Now, at National 5, you don't need to know what these structures are, but when you see these little dots attached to them, they're the same things as these little dots in the cytoplasm, and these are what we call ribosomes. Finally, we have a final arrow pointing to the middle of our cell here, and this structure is called the nucleus. So in an animal cell, you have a cell membrane, you have cytoplasm, mitochondria, ribosomes, and a nucleus. Now, this type of cell looks quite different. So this cell is a plant cell, and the easiest way to tell it's a plant cell is that it has 3 structures that are not found in the animal cell that we've just seen previously. So those 3 structures are one of these membranes on the outside, which I'll come back to in a second, these organelles here, and this big organelle in the middle. So to run through these, number one, we have the same as in an animal cell, we have a cell membrane. Now the cell membrane is the inner of the two on the outside here, and that's really important. If in an exam it points to this and it asks you to name X, you have to say cell membrane for this one. Cell membrane is the one on the inside. The one on the outside, so number two, is the cell wall. So in a plant cell, the cell wall goes around the outside, enclosing the entire cell. Now, number three is another one that's different, and it's only in plant cells compared to animal cells, and this is the vacuole. Then what we have are these two, it's pointing to here, there's some other ones in the cell as well, and these are chloroplasts. These are the things that make a plant cell green, and we'll come back to them later and their function. Now number five, again, like in our animal cell, it's pointing to these little dots that can be either free in the cytoplasm or attached to this structure. So these are ribosomes, just the same as before. Again, number six, we saw in the animal cell, this thing in the middle is the nucleus. Number seven is pointing to the white space, that's the cytoplasm. And finally, 8 is pointing to these little things here, which, as I said, kind of resemble the back of a woodlouse. So these are mitochondria. So just to highlight here, we have all of the same structures that we had in our animal cell. So we have our nucleus, ribosome, cytoplasm, mitochondria, and cell membrane. But there are now 3 separate structures that we only find in plant cells and not in animal cells. And they are the cell wall, which is on the outside, the vacuole, which is this big bit in the middle, and the chloroplasts. Okay, so now what we're going to move on to is a slightly different type of cell. So this one is a fungal cell or a yeast cell. So you can see in the exam, they might either say yeast or they might say fungal. Now, hopefully, we'll now recognise each of these parts. So number one here, we've seen this twice already. This is the mitochondria. Number two, around the outside, it'll have to be the cell wall. Number three is focusing on the nucleus. Number four is pointing to the little dots that are, this time, they're attached to the structure in the cytoplasm. So that's ribosomes. The white space is cytoplasm. And number six is pointing to the inner membrane here. So that's the cell membrane. So what you'll notice about this cell is it's basically an animal cell. It's got nucleus, ribosome, cytoplasm, cell membrane, and mitochondria. But the difference is that it also has a cell wall. So it's mainly similar to animal cells, but it has a cell wall like a plant cell around the outside. However, it doesn't have a vacuole and it doesn't have chloroplasts. Our final type of cell is this one here, and this one looks quite different. This is a bacterial cell. So again, we have number one pointing to the white space, which means it'll be the cytoplasm. Number two is pointing to the inner membrane or inner circle around the outside here, which would be the cell membrane. Now, number three is pointing to something that we haven't seen yet in any of our other 3 cell types. These are little rings, and these are called plasmids, and we'll come back to these when we do genetic engineering later in the course. Number four is pointing to the dots in cytoplasm, so these are ribosomes, we know that from before. Now, what you'll notice for number 5 is you might notice that this cell doesn't have a nucleus. It's the only one of the 4 cell types not to have a nucleus in it. Instead, its DNA, or genetic material, again, we'll come back to that in a separate topic, is just coiled in the cytoplasm. So this is what we call a bacterial chromosome. Now, you'll learn about chromosomes in a bit more detail later, but it's really important if you're ever asked what this is in the exam, you say bacterial chromosome. And finally, pointing to the outside of the cell is the cell wall. So for this one is quite different. We have our cell membrane, our cytoplasm, and our ribosomes, just like in our animal cell, but this time there is no nucleus. So we have a bacterial chromosome instead. It also contains a cell wall, just like your plant cells or fungal cells, and it also has plasmids and bacterial chromosomes. So these two are very different than the things we found in the other cell types. So now what we're going to do is we're going to focus in on these organelles. So each of those cells we've just talked through contains organelles, all those parts or structures I just pointed out, and these are needed to help the cell function correctly. To be successful in your SQE exam, you're going to need to be able to identify the names of these. So like in those diagrams we just looked at, you might be pointed to a part of it and asked what that is. But not only that, apart from knowing the names, you will also have to know the functions, so the jobs of these organelles as well. So what I would suggest you do now is that you draw this table into your jotter or onto your piece of paper. Then think back and try and tick off the cells that contain each organelle that we've gone through. So for example, think back to the plant, animal, bacterial and yeast cells that we just looked at and try and tick which ones you remember contains a chloroplast. And then do the same for each of the organelles going down the list. It's best if you could try this from memory, but if you are struggling, then feel free to go back and watch the first part of this video again. Now what I suggest doing is once that table's copied in, you copy in this table as well. So this one, we're going to learn about the functions of each of these organelles, and these are really important. This is a really common exam question. As we go through each of the following slides, I'm going to describe the functions of each of the organelles to you and which cell type they're found in. So this will allow you to mark this table that you should have already completed and to fill in this table as we go through. Pause the video here to give you some time to copy those out. Okay, so we're going to start with the cell wall. So the cell wall, remember, is the part that goes around the outside of certain types of cells. Now to fill in your first table, it's not present in animal cells, but we do find it in plant, fungal and bacterial cells. The role of the cell wall, the function, if you're ever asked, is it supports and strengthens the cell. So that's what you want to write into your table. So cell wall supports and strengthens the cell. I've also put here that it's freely permeable. We'll come back to this in area 2, transport across cell membranes, but basically what that means is that the cell wall lets anything through. It's not a solid structure that has no gaps in it. It does actually let molecules and substances through. Our second organelle that we're going to learn the function of is the cell membrane. So the cell membrane, if you remember correctly, looking at the bottom, is present in all 4 types of cell that we've looked at. The job of the cell membrane is it controls which substances or molecules can enter and exit the cell. So that's the exact definition you want to say if you're asked for the function of the cell membrane. So controls which substances or molecules, you can say either, there's a slash in between those two, can enter and exit the cell. So you want to say enter and exit. You don't want to say things or stuff instead of substances or molecules. You want to be quite scientific about this. So which substances or molecules can enter and exit the cell. The cytoplasm, remember I said, was that blank space, the white space in the middle of the diagrams. Now remember the cytoplasm is found in all 4 cell types, and the function is it's the site of chemical reactions. Now a lot of these definitions I'm going to do from now on start with site of, and that makes your definition a little bit easier. So site of chemical reactions is the function of the cytoplasm. The mitochondria, remember this diagram from our ones earlier. Now this one was found in 3 of our 4 cell types, but is not found in bacterial cells. So you need to remember it's found in animal, plant and fungal cells, not bacterial cells. And the function of the mitochondria is it's the site of aerobic respiration. It's not enough to just say site of respiration. You wouldn't get the mark for that. And we'll learn about in area 6 why that is. There's a few different locations for respiration. The site of aerobic respiration is the mitochondria. Chloroplasts. So chloroplasts are the site of photosynthesis, and they're found in plant cells, not in animal cells, not in fungal cells, and not in bacterial cells. So remember, these are the things that contain a pigment that make the plant cells green, so they are the site of photosynthesis. That's their function. Vacuole was the one that we saw in the plant cell only, not in the animal, fungal, or bacterial cells. It was the space in the middle, and this contains cell sap. So the function is it contains cell sap. Ribosomes were really important. We found them in all 4 of our cell types, and their function is they are the site of protein synthesis. So site of protein synthesis. And again, we'll come back to this in key area 3. Getting near the end now are plasmids. So plasmids we only found in bacterial cells. They can also be found in fungal cells as well, although that wasn't in our diagram from earlier. I wouldn't worry about that. The main thing you need to know is that they're found in bacterial cells, and they are circular loops of DNA or genetic material. These ones are less commonly asked about in terms of function. They have a whole topic to themselves later, so don't worry too much about this one at the minute. Finally, we have our nucleus. So nucleus was found in 3 of our 4 cell types, so animal, plant, and fungal, and was not found in bacterial. The nucleus definition or function is really, really important. Controls some cell activities. The most important thing here is you never say that it controls all cell activities. There are other parts of the cell that are involved in some cell activities and the control of them. So you need to say controls some cell activities for this one. And remember, it's not found in bacterial cells. Okay, before we move on, that was all our functions, so your table should now be filled in. We need to just focus on the cell wall for a moment. So the cell wall we saw in the diagrams before, that the cell walls found in plant cells, fungal cells, and bacterial cells. Now, the important thing here is it's not found in animal cells, but you also have to know about a difference that we find between the cell wall in plant cells and the cell wall that's found in fungi and bacterial cells. Now, this is a really, really common exam question, but it's one that a lot of people forget to revise in KA1. So the cell wall in plant cells, you need to know, is made up of a substance called cellulose. So cellulose is the substance that makes up plant cell walls. However, the cell wall in fungi and in bacterial cells is still there, but it's made up of a different material or different component. So you just need to know that the plant cell wall is made of cellulose, whereas the cell wall in fungi and bacterial cells is made up of something else, something different. So please make a note of that. Okay, a quick revision tip since we've done most of the topic. The best way I've found to practice and to learn the parts of their cell, their structures and their functions is to try and draw them out and label them from memory. So get a blank piece of paper, try and draw it out, try and label all the parts you can, try and write the functions, and then open your notes, mark them, mark your drawings using your notes, and use a different colour of pen where possible. Make sure you know and fill in those areas that you didn't get correct with that different colour of pen. And then a day or two later, try the whole thing again, repeat it. Do not look back at the old one that you did a couple of days before, and do not look at your notes right before you attempt to draw it. If you look at your notes, you're just using your short-term memory. It's not going to help you remember it in the long term. What I would do for the next wee while, over the next couple of weeks as we focus on this topic, is just to keep doing this over and over again until you can easily draw all 4 cell types, label all of the parts and write their functions, and then you know you're ready to move on. Okay, just to do a quick knowledge check as well, can I get you in your notes, or just even to think about it, to match up the following organelles with their correct function? So you don't have to write all of this out, you could just write 1, 2, 3, 4, 5, 6, 7, 8, and then tell me what function is beside each one. Pause the video here, work this out, and then we'll go over the answers in a second. Okay, so for number 1, for cytoplasm, we should know that that is the site of chemical reactions. So it must be F. So 1 goes with F. For number 2 is the ribosomes. The ribosomes were the site of protein synthesis. So H. 3 is the chloroplasts. The chloroplasts are the site of photosynthesis. So G. 4 for nucleus controls some or most of the cell's activities. So D. 5, mitochondria, site of aerobic respiration, so E. 6, cell wall, supports and strengthens the cell, so C. Cell membrane controls entry and exit of substances into and out of the cell, so A. And then 8, vacuole, it contains cell sap, so B. If you found that difficult to do without using your notes, go back and read your notes again, watch the rest of the video again, and then give this another try. You need to get to the point where not only can you match these up really easily, but you can also tell me these from memory. Okay, so now what we're going to do is we're going to move on and we're going to look at microscopes and about how we can look at cell structure and ultrastructure. So cells do vary in size, but most of them are small enough that you need to use a piece of equipment called a microscope to study their structure. Now, we normally measure their size in terms of micrometers. Now, you'll have heard of millimeters before, you'll have heard of centimeters, but you might not have heard of micrometers. Micrometers is used for much smaller sizes. So micrometers uses this little U with a tail at the front and then M. So like millimeters is mm, little m, micrometers is this fancy U, and then M is what you use to describe it. Now, there's 1,000 millimeters in a meter, hopefully you know that, and there's actually 1,000 micrometers in a millimeter. Now, you don't need to remember this at the moment, but we'll come back to this soon when we do some calculations. Before we do, usually what you would get to do in this topic if you were in school is you would get to look down microscopes and look at different types of cells. So I'm just going to show you some pictures of those so that we can match those up to what we've seen in the diagrams already. So cheek cells are a type of animal cell. So animal cells, because we can see here it's got a nucleus and we can see it's got cytoplasm and a cell wall, a cell membrane, sorry. Now with this one, it's quite important that we point out that you cannot see everything in the cell, like the diagrams we looked at before. We need to zoom in a lot closer to be able to see a lot of those structures that are a lot smaller, like the mitochondria and the ribosomes. But you can see the nucleus, you can see the cell membrane, and you can see the cytoplasm, just like you can here. Red onion cells are a type of plant cell. So for this one, you can see the cell wall and cell membrane going around the outside, and in red onion cells, the cytoplasm is dyed a red colour. Yeast cells, these ones here, you can see their cell membrane and cell walls, and you can see their nucleus as well. Kabomba is a type of plant cell. So here you can see the cell membrane, cell wall, nucleus, and you can also see the chloroplasts that are green in colour as well. Okay, so what we're going to do now is we are going to look at working out the size of a cell. Now this final type of question they tend to ask in the exam in this topic is to do with calculating sizes of cells. It's a really common type of question. It's a really common type of calculation. You may need a calculator to do this. If you're good at mental maths, you should be fine, but you can always use your phone to work out these as well. Now in any questions like this, they will tell you the field of view of the microscope and they will show you the number of cells. Now just to explain this in a bit more detail, this is pretending that you are looking down the microscope at a bunch of cells. Now these don't look very detailed, but these are meant to be cells, different types of cells. So as we look down the microscope, you can see there's 1, 2, 3, 4, 5, 6, 7, 8 cells down here. Now the field of view is basically the distance side of this. So the field of view here is 2 millimeters. Now it's 2 millimeters across, but because it's a circle, it's 2 millimeters in any kind of orientation along here. So 2 millimeters over here, 2 millimeters down here. Now what they would ask you in the exam is to calculate the size of one of these cells, so the length of the cell. So if I want to calculate the length of one cell and I'm pretending that all of them are basically the same size to start off with, you use this calculation. So field of view, which they will always give you, divided by the number of cells. Now you need to be really careful here because number of cells does not mean total number of cells. It means the number of cells that are sitting side by side. So what we would do for this example here, if I wanted to work out the size of one cell, would be I'd take the field of view, which is 2 millimeters, and I would divide it by the number of cells that are sitting side by side. So 1, 2, 3, 4, 5. So 2 divided by 5, which gives you 0.4 millimeters because it was 2 millimeters to start with, so we must include our units at the end. So what this means is that each cell here roughly measures 0.4 millimeters in length. Now we're going to try a few different examples of this. Now I've given you the answers here, but I'm going to explain how I got to each of these. So the diagram below shows a group of cells as seen under a microscope. The field of view was 2 millimeters in diameter. So even though it's not got the wee bit at the top, it's telling me in the question that 2 millimeters is the field of view. It says calculate the average length and height of the cells. So it's 2 marks, this one. They wouldn't usually do this, but this is just to get you practice. So we're going to do length first. So if I want to calculate the length of this a cell in this field of view, I would take field of view divided by number of cells. So field of view is 2 divided by the number of cells. Now I'm wanting to work out the length, so I count across the way. 1, 2, 3, 4, 5. So I do 2 divided by 5, and that gives me 0.4 millimeters as my final answer. However, to calculate the height, it's not the same answer, because if I take 2 and I divide it by the height, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 cells going up and down the way. So that would mean 2 divided by 10, which gives 0.2 millimeters. So that's my 2 answers there. Now that's the most basic type of calculation, is them asking the length or height of a cell. And you don't have to do any conversion. However, they can make this slightly more challenging. So this is the same type of question. I'm just focusing on length now, but you can see I've added a little bit to the question. So this time, field of view, still 2 millimeters, but calculate the average length of the cell in micrometers. So what this means is, even though my field of view is measured in millimeters, I want my final answer, instead of being in millimeters like it was here, to be in micrometers. Now, in an exam question on this, they would tell you the conversion. They will always tell you this. So 1 millimeter equals 1,000 micrometers. So you do the calculation in the exact same way as we did with the easier questions, and then you just add a tiny bit at the end. So to work out the length, just like before, you would take your 2 millimeters across, you would divide it by 5 because there's 5 cells, and that would give you 0.4 millimeters. Now, to get this into micrometers, we need to convert it, and we use the information we were given here, that 1 millimeter equals 1,000 micrometers. So all you do is you take your answer and you times it by 1,000 to get it into micrometers. So my final answer for this one is 400 micrometers. And remember, to write micrometers, you use that little U with the tail at the front, and then just a small m. So hopefully that makes sense. So it's the exact same calculation, but you just, to convert it into micrometers from millimeters, you just times by 1,000. So what we're going to do now is we're going to try 3 example questions. Now these are a mix of some easier ones and some slightly harder ones. I want you to pause the video here, write out your answers, make sure you do show your working as well, and then come back to the video when you're ready to go through the answers. Okay, so for the first one, it asks you to calculate the length of the average cell in picture A. Now it says length. The first thing we need to work out is our field of view. So our field of view is 12 millimeters. Then what we need to do is we need to divide 12 millimeters by the number of cells. And then we're working out the length. So 1, 2, 3, 4, 5, 6. We don't count these ones at the side. So it's 6. So 12 divided by 6. And that answer should have given you 2 millimeters as the final answer. Remember, 2 by itself would not get your mark. You have to include your units. Number 2, calculate the height of the average cell in picture B. So this time our field of view is 28 centimeters. Now we need to realise that the final answer is now going to be in centimeters, not millimeters. So 28 is our field of view, and we divide it by the number of cells. Now, because we're doing height, we want to count up and down the way. So 1, 2, 3, 4, 5, 6, 7. So 28, which is the field of view, divided by 7, and our answer should have been 4 centimeters, because it was centimeters at the start. Now finally, this is the challenging one. For number 3, calculate the length of the average cell in picture C in micrometers. And again, it's given us that information that we need to do the conversion. So this time what we do is we take the field of view, which is 4.4 millimeters. We divide it by the number of cells, and because we're doing length, we count across the way. So 1, 2, 3, 4. So 4.4 divided by 4, just like normal. That means our answer at the minute is 1.1 millimeters. However, that is not our final answer, because we want our final answer to be in micrometers. So we times by 1,000, and that gives us 1,100 micrometers, which is our final answer. Okay, so a good way to practice this at home is to make your own. So you can draw basically a version of this diagram. You come up with a field of view. You can even use a ruler to measure it. Draw however many cells you want, and then you can decide whether you want the final answer to be easy, so in millimeters, or whether you want to change the final answer to be in micrometers, so you have to do the conversion. This is one of the best ways to practice this, is to make up your own sheets of them, you work out your marking scheme, and then you can keep this for later in the year to help you revise as well. Okay, so just to run through what we've gone through today, we've learned about the main cell structures, organelles, and their locations within the cell, and we've learned about the basic functions of each organelle as well. Hopefully now you can list all of the organelles in National 5, and you can briefly explain the function of each of those organelles. You should be able to label the organelles in the 4 different cell types and be able to recognise those 4 different cell types as well. And finally, you should be able to complete some numeracy calculations on the sizes of cells. Now, just before you go, just to make sure you've understood everything, I think you should try the plenary questions. So again, pause the video, write down your notes or think about your answers, and then we'll go through the answers before you go. So first one is, what is the function of the ribosome? Second one, what is the function of the nucleus? Third one, give the 3 structures that are found in plant cells but are not found in animal cells. 4, describe the difference between a plant cell wall and a bacterial or fungal cell wall. So pause the video here, write down your answers, and then we'll go through them. Okay, so for the first one, what is the function of the ribosome? It should have been site of protein synthesis. Function of the nucleus, controls some cell activities, not all. Give 3 structures that are found in plant cells but not in animal cells. This is a really common type of question that they'll say, name some structures that are found in this type of cell but not in this type of cell, which is why it's so important that you can picture those diagrams and you can draw them out. So for structures that are found in plant cells but not in animal cells, it's the vacuole, chloroplast, and cell wall. And finally, describe the difference between a plant cell wall and a bacterial cell wall. Now, a big important thing here is when you get asked to do the difference between 2 things in biology, you'll need to make sure you mention both of them. It's not enough just to say plant cell walls are made of cellulose, because you've not mentioned bacterial or fungal cell walls, which are in the question. So your perfect answer for this would be the plant cell walls are made of cellulose, whereas bacterial or fungal cell walls are made of something different. So main thing here is if you get a question that asks about difference or comparing, you say one is like this, whereas the other one is like this, and that means it's definitely a comparison. So remember, plant cell walls are made of cellulose, whereas bacterial or fungal cell walls are made of something else. Okay, so well done for completing your first National 5 lesson. If you've got any questions at all, either watch the video back again, see if that helps you answer them, or if you still have any questions whatsoever, please let your teacher know. Well done.